Hello everyone, my name is Ruby and welcome to my channel. Today, I'll be giving a brief introduction to the technique of DNA cloning. DNA cloning is a technique that is used to generate lots of copies of our desired gene. This is useful when we need many copies of our gene for analysis, for gene therapies, or if we would like to mass-produce proteins that are encoded by our target gene. The way it works is quite intuitive. We know that bacteria replicate quite fast, and that they could take a foreign DNA in the form of a plasmid, a circular double-stranded DNA. If we insert our target gene inside a vector, a plasmid designed for DNA cloning, and put it into bacteria, the bacteria could mass-produce our target genes, and even its encoded proteins as it grows and replicates. To insert our gene into our vector, we need to cut both our gene and vector with restriction enzymes that recognize specific sites on DNA and cut at certain bases. Often, the enzymes generate sticky ends, which are 5' or 3' overhangs on our gene and our vector. This allows our target gene to base pair with our vector, allowing its successful insertion. In our diagram, the restriction enzymes EcoR1 and KPN1 bind to and cut at their specific restriction sites on both our vector and our gene. The orange overhang generated by EcoR1 could only bind to the orange overhang it generated on the gene. The red could only bind to the red. This process of using two different enzymes to generate non-compatible overhangs is called double digest. This helps our gene insert in the correct orientation and helps prevent our vector from sticking back onto itself. This is important in protein productions, since inserting our gene in the opposite direction could prevent our target protein from being produced. After we have our gene bound to the vector, the next step will be to glue them together using the enzyme called DNA ligase. DNA ligase joins the 3' hydroxyl and the 5' phosphate groups on our gene and our plasmid, pasting them together to form an unbreakable recombinant plasmid. Once we have a recombinant plasmid, we need to put them into bacteria in a process called transformation. A common way of transformation is heat shock. First, we incubate our bacteria in calcium chloride to make them competent, to help our bacteria get closer to our plasmids. Then, we briefly shock them with a higher temperature of around 42 degrees Celsius. This changes the fluidity of the bacterial cell membrane, creating pores that allow them to take in our plasmids. However, not every single bacterium wants to take in our recombinant plasmids. To only select for those that contain our plasmids, those that are successfully transformed, we include an antibiotic-resistant gene inside our plasmid. After placing our bacteria on an agar plate containing antibiotics, those that took up our plasmid will be protected by the antibiotic-resistant gene and survive. Those who did not take up the plasmids will be killed by our antibiotic. How do we know that all these successfully transformed bacteria will contain our target gene at the right place in their plasmids? It is possible that our vector ligates back onto itself, meaning that it does not contain our gene. It is also possible that our gene is inserted into the opposite direction, or at the wrong location. 3 common methods to verify successful insertion of our gene will be Colony PCR, restriction digest, and sequencing of our recombinant plasmid. Colony PCR is a fast and cost-effective method that directly uses our bacterial plasmid as a template for the PCR reactions. We simply pick up a bacterial colony, add them to our PCR reaction mix, run the amplified fragments on our gel to separate them by size, and then analyze the bands. The heat of the PCR reaction releases our plasmids from the bacteria and linearizes them for the reaction. Through choosing specific primers that amplifies different segments in our PCR reaction, this technique allows us to identify the presence, size, and orientation of our target gene or insert. To determine the presence of our insert, we choose insert-specific primers that recognize and amplify only our insert. This means that if the plasmid contains our target gene, the gene would be amplified through PCR, and we would see a clear visible band on our gel. However, if the plasmid does not contain our gene, there is nothing for our primers to recognize and amplify, so we would not see anything on the gel. We could also determine the size of our insert by choosing flanking region-specific primers that recognize regions flanking our target. If our gene is present, our primers would amplify both the flanking regions along with our gene, resulting in a larger fragment. If our gene is absent, our primers would only amplify the flanking regions, resulting in a significantly smaller fragment. Since the smaller fragments run faster on the gel, our faulty plasmid without the insert would be much lower on the gel compared to our plasmid containing the target gene. To determine the orientation of our insert, we could use orientation-specific primers that recognize a region inside of our insert and a region flanking our insert. If our gene is in the correct orientation, the primers could successfully amplify our gene and we would see a clear visible band. However, if our gene is in the opposite direction, the primer inside of our insert would also be in the opposite direction, and our gene would not be amplified, resulting in no band on our gel. A more traditional way to verify successful gene insertion into plasmid is restriction digest. Restriction digest involves using specific restriction enzymes to cut our recombinant plasmid into fragments, running the fragments on the gel to separate them by size, and then analyzing their banding patterns to gain information about the size and orientation of our insert, as well as the identity of our plasmid. Restriction digest allows us to identify the size of an entire plasmid, our insert, and the remaining backbone. To know the size of an entire plasmid, we linearize our plasmid by cutting it at a single site using a restriction enzyme, and then running it on a gel. The band's location on the gel would tell us the size of our plasmid, which is around 6,200 base pairs. Similarly, to know the sizes of our insert and our backbone, we use two restriction enzymes to cut around the insert, resulting in two fragments corresponding to our insert and the backbone. The size of the insert and backbone fragments could also be visualized on the gel. In this case, our insert is 1,200 base pairs, and our backbone is 5,000 base pairs. To determine the orientation of our insert, we use a restriction enzyme that cuts at both outside of our insert and at an off-center location inside of our insert. If our gene is in the wrong orientation, the restriction enzyme would cut at a different location within our insert, and it would generate fragments of different sizes compared to our desired insert. By comparing the fragment sizes on our gel, we could tell if our gene is inserted in the right direction. Finally, we could use restriction enzymes to determine the identity of our plasmid through a technique called plasmid fingerprinting. By choosing a specific restriction enzyme to cut our recombinant plasmid into multiple segments, separating those segments by size on the gel, and then comparing our band pattern to the expected plasmid pattern on the website, we could determine if our plasmid is indeed the one that we want. We usually use more than one restriction enzyme for further confirmation of its identity. If we want to use our gene to synthesize proteins, we must make sure that it is inserted at a correct sequence within our plasmid, so that it will be in the right reading frame for translation. In this case, we would send our purified recombinant plasmids for sequencing. Common methods of sequencing are next-generation sequencing, such as the Illumina platform, and the traditional method of Sanger sequencing. For simplicity, I will briefly go over the theory behind Sanger sequencing. Sanger sequencing is really similar to PCR, in that we would first denature and separate the DNA strands, anneal a primer to our DNA template, and then use a DNA polymerase to elongate and synthesize our new DNA strand. However, the major difference in Sanger sequencing is the use of terminator bases conjugated to a fluorophore. Once these terminator bases are incorporated into the chain, they stop the chain from elongating, resulting in fragments of varying lengths. These fragments will be separated by size through gel electrophoresis in a capillary tube. and a terminator base on the smallest fragment will be detected first. Since each type of terminator base is conjugated to a different fluorophore, we could record the different colors, translate these colors into bases, and eventually get the exact sequence of our plasmid. By knowing that our bacteria contains our plasmid with our target gene successfully inserted, we could be more confident that the copies of genes produced by our bacteria through DNA cloning will be the ones that we want. Thank you so much for watching my crash course, and I hope this video could help you understand more about the technique of DNA cloning. Feel free to comment below for any questions or concerns.